So now that you hopefully have generated your library, what you would like to do, and that you have sequenced, of course, what you would like to do is to analyze your sequences. So here is the analysis pipeline and the file formats that are associated to it. So initially, the huge amount of data that is produced by the NGS instruments can be really scaring. We are talking today about millions of reads sequenced, and these need to be carefully handled. So this is why there are a huge number of algorithms, of softwares, of tools and different methods that are constantly developed in order to ease that process. So you need to define first a streamlined pipeline in order to facilitate that data analysis, and in order also to be sure that you are interpreting correctly your biological question. So you have here an example of the classical key steps of a pipeline of analysis. I simplified it here with a number, restricting it to a certain number of files that are generated at each step. So I really only focused here on the key files that are involved for the sake of space and time, although I highlighted here one particular step after the generation of the BAM files, because this step has a great importance in all the following steps, both for genomic coverage and for variant calling. And we will also see the files that are required as input in the following slides. And we will talk a bit about some of the tools or softwares that are used and on which you can rely to generate or exploit these files. So the basic steps are first to take the raw sequencing reads out of your platform, then align them on a reference genome, then calculating the genomic coverage, then identifying large structural variations or copy number variations for genes or chromosomes, or variant calling for SNPs and indels. And then, of course, the biological interpretation of your datasets. So the first thing that you get as an output is a FASTQ file format. So this is what is the most common format. So it could be also an SRA, which is a binary format, but this is rather used for public data online, and we will be seeing this in the practical part. So what are the major characteristics of the FASTQ file? So in fact, if we go back first to the Sanger sequencing, a real breakthrough of the Sanger sequencing is that not only it provided the sequence of a DNA fragment, but it associated to the basis of each fragment a quality value or a quality score, and this was called the Fred quality score. So basically, if you have a high score, then you have a higher quality of your basis. So thanks to the FASTQ files, the same concept can be maintained, because in these files you have, despite the huge amount of data generated, a calculation of the quality of each of the bases, and this is reported in the file. So you have not only the sequence of your read, but also the quality that is associated to it in a single file. So what do they contain, these FASTQ files? In fact, they contain millions of rows, of course. Each read is represented by a group of 4 lines. So the first line that starts with an @ sign. So each read basically would be identified by that @ sign, because to that sign is associated the sequence name, which is a unique identifier for the read. Then you have the sequence itself. Then you have a plus sign that was first used to repeat again the name of the reference sequence. You have an example here indicated by the red arrows. And then you have a quality. So the string of quality values, for which you have one character that is associated to one base, and each character represents a quality using a code that we call an ASCII code, but we don't have to go into the details of it. Just what you need to know is that these could be translated by a number of downstream programs, and be careful because the scale of these quality score could be different between different platforms. So, for example, if you have values higher than 90, then this would be an Illumina score, etc. So, FastQ files have to be quality controlled. So this is the first step to perform. This is performed by what we call a FastQC, or fast quality control step, which generates an HTML file with all the information that is indicated on the left-hand side here of the screenshot. And in the graph that is indicated here, what we have is the basis or the nucleotides of the read that are indicated in the x-axis, and the quality score that are associated to it on the y-axis. So here you can see as an example the Illumina encoding indicated here. So the graph shows the quality score of each basis of the read, so across all the length of the read. So this is an example of a very good quality reads. Now you have a case where the sequences are of relatively poor quality, especially towards the end of the read here. So in this case, where the quality control is generally followed by another step, which is called trimming. And in this step, the final basis of the reads that are showing poor quality can be discarded in order to improve the mapping. Or we could also improve it by removing bad quality reads if they are in negligible proportion. The trimming at this stage would make sense because the reads are of poor quality, and this is especially true for de novo sequencing, so be careful if you are doing de novo sequencing, because for these kind of sequencing, the genome assemblies can really be improved by data cleanup. In this second step, the required input file are, of course, at this stage, your FastQ file that have been trimmed or not. Then you would need a reference genome in a basic FASTA format. So this is the .FASTA or .FA that would contain the whole sequence of the genome you are interested in the FASTA format. The quality of this reference sequence would depend, as we saw it previously, on the status of finishing or completion of your genome. So if you do not have such a reference, maybe you would need to perform a de novo sequencing before. On the top of these data files, some programs would require that for a faster access to the data, you would need what is called a companion file, which is the index file. So here we have indicated the .fai file as an example. So this is an indexed file, and once the index is created for a given FASTA file, then this would allow to easily extract specific sequence from your datasets, like for example whole chromosomes or specifically targeted regions or something like that. Then you would also need to have a file that is called GFF file, that stands for General Feature Format, or GTF, that stands for General Transfer Format. And recently people are using more the GFF3 format. So this file contains the information about each feature that is annotated in a genome, and we will be seeing an example of this. So it associates also the position of this feature in the genome, etc. Then you would also need algorithms to align or map these reads to the genome. And maybe the currently and most popular used ones are BWA, or also a variant called BWA-MEM, MEM, and also Bowtie, that are open source and are using what is called a Burrows-Wheeler transform that improved tremendously the field of reads mapping, because it contrasted with the previously known seed-based approach that is basically cutting the genome and the reads into small seeds and then mapping them back, whereas the Burrows-Wheeler transform would look for reads identity, character by character, and this is what makes them really faster algorithms. So your output file formats would be a .sam and a .bam that are generated by these algorithms. In fact, of course, they all have, BWA and Bowtie have their, advantages and drawbacks as all other algorithms, but they are quite often used nowadays because they are fast. Others are designed for de novo sequencing, so you need to carefully look at the ones that you use before using them. So SAM is the first output format. We will be seeing characteristics of it, and BAM is its binary counterpart. So first, let's have a look at the GFF3 format. So this format contains 9 columns and other optional additional information. So the first one is a sequence ID, which is the name of the chromosome or the scaffold. You have here an example that is taken from Ensemble annotation. Second would be the source, which is the name of the program or source of this feature. Be careful because a point here means that this is an empty value. So 3 would be the type of the feature. So you have mRNA or exon here. 4 and 5 are the start and end here of the feature with a sequence numbering starting at 1. So this is something you need to take care of while dealing with the different file formats, because some have a 0-based counting and some others have a 1-based counting. So this is something that the tools or softwares knows how to deal with. The other column would be a score that is associated, then the strand for plus or minus, and then a field called phase, which would be either 0, 1, or 2, and it indicates, in fact, if the first base is in a codon or not. Is the first base of a codon or not. Then you have attributes that are different characteristics. And I would invite you to go into more details by reading the GFF documentation that you can find available on the internet. Now, the SAM and BAM file format are formats that could contain information about the mapped and unmapped reads. The SAM stands for Sequence Alignment Map and is really helpful per se, but in that format is really heavy, so it's usually used as a BAM format, which is its binary counterpart. It can contain extensive information about the read, its properties, its alignment to reference sequence, etc. So it is a format that is really widely adopted today. In fact, that file can store the mapped reads, the unmapped, the flagged one, the QC failed reads, etc., etc. So you have a whole bunch of information that is related to it. This is a file that contains a header section and an alignment section. I won't be covering the header section because it contains different kind of lines, different kind of information, and I would invite you to have a look at the websites I'm indicating here for more details. The most important section is, of course, the alignment section, which contains different information about the read. So the first one is what is called the Q name, which is the read name. Then you have a flag, which is kind of a bitwise comparison of flags, meaning that each bit corresponds to an explanation about the alignment. So, for example, a bit of 2 would indicate that each segment is properly aligned, 4 would mean that the segment is unmapped, etc., etc. Then you have our name, that is the reference sequence, etc., and others. And maybe one of the most important ones would be the cigar string, which is a sequence of numbers and letters. And we will be covering this in the next video because this kind of string and the other information is really useful for that format.